So, in the late 1950s, there was coding problem. So the question was, how is the information in a linear sequence of nucleotides in RNA translated into the linear sequence of a chemically quite different set of units, the amino acids in proteins? And now the code is deciphered. As you can see, these are the genetic code. So once an mRNA has been produced by transcription and processing, the information present in its nucleotide sequence is used to synthesize a protein. The conversion of the information in RNA into protein represents a translation of the information into another language that uses quite different symbols. Moreover, since there are only 4 different nucleotides in mRNA and 20 different types of amino acids in a protein, this translation cannot be accounted for by a direct one-to-one correspondence between a nucleotide in RNA and an amino acid in protein. The sequence of nucleotides in the mRNA molecule is read in consecutive groups of 3. RNA is a linear polymer of 4 different nucleotides, so there are 64 possible combinations of 3 nucleotides. However, only 20 different amino acids are commonly found in proteins. Either some nucleotide triplets are never used, or the code is redundant, and some amino acids are specified by more than one triplet. The second possibility is, in fact, the correct one. And each group of 3 consecutive nucleotides in RNA is called a codon, and each codon specifies either one amino acid or a stop to the translation process. This genetic code is used universally in all present-day organisms. Although a few slight differences in the code have been found, these are chiefly in the DNA of mitochondria. Mitochondria have their own transcription and protein synthesis systems that operate quite independently from those of the rest of the cell, and it is understandable that their tiny genomes have been able to accommodate minor changes to the code. In principle, an RNA sequence can be translated in any one of 3 different reading frames, depending on where the decoding process begins. However, only one of the 3 possible reading frames in an mRNA encodes the required protein. So a special signal at the beginning of each RNA message sets the correct reading frame at the start of protein synthesis. The codons in an mRNA molecule do not directly recognize the amino acids they specify. The group of 3 nucleotides does not bind directly to the amino acid. Rather, the translation of mRNA into protein depends on transfer RNAs, which are about 80 nucleotides in length. So, there are many RNA molecules can fold into precise 3-dimensional structures, and so are tRNA molecules. So 4 short segments of the folded tRNA are double helical, producing a molecule that looks like a cloverleaf when drawn schematically. The cloverleaf undergoes further folding to form a compact L-shaped structure that is held together by additional hydrogen bonds between different regions of the molecule. 2 regions of unpaired molecules situated at either end of the L-shaped molecule are crucial to the function of tRNA in protein synthesis. One of these regions forms the anticodon, a set of 3 consecutive nucleotides that pairs with the complementary codon in an mRNA molecule. The other is a short single-stranded region at the 3' end of the molecule. This is the site where the amino acid that matches the codon is attached to the tRNA. So, the genetic code is superfluous. So this implies that either that there is more than one tRNA for many of the amino acids, or that some tRNA molecules can base pair with more than one codon. In fact, both situations occur. So some amino acids have more than one tRNA, and some tRNAs are constructed so that they require accurate base pairing only at the first 2 positions of the codon and can tolerate a mismatch or wobble at the 3rd position. This wobble base pairing explains why so many of the alternative codons for an amino acid differ only in their 3rd nucleotide. In bacteria, wobble base pairings make it possible to fit the 20 amino acids to their 61 codons with as few as 31 kinds of tRNA molecules. The exact number of different kinds of tRNAs, however, differs from one species to the next. For example, humans have nearly 500 tRNA genes, and among them, 48 different anticodons are represented. So, tRNAs are covalently modified before they exit from the nucleus. Eukaryotic tRNAs are synthesized by RNA polymerase III. Both bacterial and eukaryotic tRNAs are typically synthesized as larger precursor tRNAs, which are then trimmed to produce the mature tRNA. In addition, some tRNA precursors from both bacteria and eukaryotes contain introns that must be spliced out. This splicing reaction differs chemically from pre-mRNA splicing. So rather than generating a lariat intermediate, tRNA splicing uses a cut and paste mechanism that is catalyzed by proteins. So trimming and splicing both require the precursor tRNA to be correctly folded in its cloverleaf configuration. Because misfolded tRNA precursors will not be processed properly, the trimming and splicing reactions serve as quality control steps in the generation of tRNAs. So, all tRNAs are modified chemically. Nearly 1 in 10 nucleotides in each mature tRNA molecule is an altered version of a standard G, U, C, or A ribonucleotide. Over 50 different types of tRNA modifications are known, and some of the modified nucleotides, most notably inosine, produced by the deamination of adenosine, affect the conformation and base pairing of the anticodon and thereby facilitate the recognition of the appropriate mRNA codon by the tRNA molecule. Others affect the accuracy with which the tRNA is attached to the correct amino acid. So, we now consider how each tRNA molecule becomes linked to the one amino acid in 20 that is its appropriate partner. Most cells have a different synthetase, aminoacyl-tRNA synthetase enzyme for each amino acid. that is 20 synthetases in all. And one attaches glycine to all tRNAs that recognizes codons for glycine. Another attaches alanine to all tRNAs that recognize codons for alanine, and so on. And many bacteria, however, have fewer than 20 synthetases, and the same synthetase enzyme is responsible for coupling more than one amino acid to the appropriate tRNAs. In these cases, a single synthetase, places the identical amino acid on two different types of tRNAs, only one of which has an anticodon that matches the amino acid. So a second enzyme then chemically modifies each incorrectly attached amino acid so that it now corresponds to the anticodon displayed by its covalently linked tRNA. The synthetase catalyzed reaction that attaches the amino acid to the 3' end of the tRNA is one of many reactions coupled to the energy-releasing hydrolysis of ATP, and it produces a high-energy bond between the tRNA and the amino acid. The energy of this bond is used at a later stage in protein synthesis to link the amino acid covalently to the growing polypeptide chain. So, an amino acid is activated for protein synthesis by an aminoacyl-tRNA synthetase enzyme in two steps. The energy of ATP hydrolysis is used to attach each amino acid to its tRNA molecule in a high-energy linkage. The amino acid is first activated through the linkage of its carboxyl group directly to AMP, forming the adenylated amino acid. The linkage of the AMP, normally an unfavorable reaction, is driven by the hydrolysis of the ATP molecule that donates the AMP. So without leaving the synthetase enzyme, the AMP-linked carboxyl group on the amino acid is then transferred to a hydroxyl group on the sugar at the 3' end of the tRNA molecule. This transfer joins the amino acid by an activated ester linkage to the tRNA and forms the final aminoacyl-tRNA molecule. So there are two major classes of synthetase enzymes. One links the amino acid directly to the 3' OH group of the ribose, and the other links it initially to the 2' hydrox OH group. In the latter case, a subsequent transesterification reaction shifts the amino acid to the 3' position. So, several mechanisms working together ensure that an aminoacyl-tRNA synthetase links the correct amino acid to each tRNA. Most synthetase enzymes select the correct amino acid by a two-step mechanism. The correct amino acid has the highest affinity for the active site pocket of its synthetase and is therefore favored over others. So in particular, amino acids larger than the correct one are excluded from the active site. However, accurate discrimination between two similar amino acids is very difficult to achieve in a single step. A second discrimination step occurs after the amino acid has been covalently linked to AMP. When tRNA binds, the synthetase tries to force the adenylated amino acid into a second editing pocket in the enzyme. The precise dimensions of this pocket exclude the correct amino acid while allowing access by closely related amino acids. In the editing pocket, an amino acid is removed from the AMP or from the tRNA itself if the aminoacyl-tRNA bond has already formed by hydrolysis. This hydrolytic editing is analogous to the exonucleolytic proofreading by DNA polymerases. So, the tRNA synthetase must also recognize the correct set of tRNAs. And most tRNA synthetases directly recognize the matching tRNA anticodon. These synthetases contain 3 adjacent nucleotide binding pockets, each of which is complementary in shape and charge to a nucleotide in the anticodon. For other synthetases, the nucleotide sequence of the amino acid accepting arm, the acceptor stem, as I mentioned a while ago, this is the acceptor stem, is the key recognition determinant. In most cases, however, the synthetase reads the nucleotides at several different positions on the tRNA. So, okay. This is the end of the video. Thank you for watching.